All right, so you haven't missed much. I apologize. I went off the sharing. So just what I wanted to do was to talk about how much of you is viral. And then I showed you this slide where I said 10 to the power 31 numbers of viruses are estimated to be on Earth. I think it is a pretty underestimate of what actually occurs. Just to make I learned. So I also said that we would love a viral-free world, but if you look at viruses, they are everywhere. So, for example, a teaspoon of seawater contains 100 million viruses, and they do enormous amount of work. So you can see that most of the viruses that are around us and within us is bacteriophages, which infect bacteria. And the key virus that we see is the virus that infects the ancient unicellular organisms called which are the producing photosensitive viruses that the ancient virus that will cause photosynthesis and store the energy. Now, viruses, when they enter these bacteria, stop this process with that infection of breaking down of the energy, and they rather make them store that and stop the usage of the energy by them, thereby releasing oxygen. So if you think of the breath you took just now, 50% of that is actually virally contributed. That, in other words, they promote photosynthesis in the bacteria in the ocean, and therefore half of what we breathe is estimated to be virally produced. You can also see that the viruses control the populations of organisms from this complex interaction that we see in this cartoon, and that through that action, they control not just the number of bacteria in the ocean, but also the other organs. So as you see, the monocellular organisms in the ocean is how life starts and their food chain starts, and the viruses actually influence that process. So if you want to see whales in the ocean, that requires the photosensitivity and the photosynthesis that occurs from the unicellular organisms, and viruses actually promote it, arrest the energy usage in that organism, thereby giving more energy to the next level, planktons and others, and the small organisms, which then feed the fish and which feed the larger animals like the whales. So it's fascinating that life as we see it is dependent on viruses and the viral bacteriophage organisms that we call as bacteriophages. So every breath we take seems to be contributed to by the viruses. If you look at the human microbiome, we know that there is quite a lot of bacteria in our gut, and we know that there are at least 100 billion per gram of feces of bacteria in our feces, 30 trillion cells compared to 38 trillion bacteria. So the body contains 30 trillion cells, and you can see that there are 38 trillion bacteria, which is much higher number, and probably 10 times that number of viruses or viral particles, most of them viral phages, which infect the bacteria within the gut. What do they do? We don't know much about that. So that's the surprising part about it. These viruses that we know seems to be pretty fixed. So here you can see in the gut virome, you can see that the types of the organisms or the viruses that we have seems to be pretty fixed. So in infancy, there is one set of viruses and bacteriophages, whereas in the adult life for each individual, there seems to be a fixed number of gut viromes that are of particular type. And what they do, we don't know very well. However, in this April, I found this article from the journal Microorganisms, and I have redrawn that to make it simple. You can see the concept of the human microbiome, including bacteriophages, nematodes, and the bacteria, and how they interact with our own immune system to contribute to the immunity and sometimes autoimmunity. So Dr. Shubhankar at the beginning alluded to how autoimmunity by viruses can give rise to, say, type 1 diabetes and other autoimmune conditions. And so you can see that the gut microbiome, or virome, if you want to be more specific about viruses, contributes to or interferes with human inflammation, sometimes positively, sometimes negatively. Now, when I researched further about viruses, I was also reminded of this cartoon that we learn in our second MB and third MB, where we learn about the various ways in which a virus could transfer and give a new DNA sequence to a cell. We learned that about in how bacteria suddenly become resistant to an antibiotic by a new gene that had been paged, which is a viral particle. But you can also appreciate here that there are multiple methods by which gene transfer could occur horizontally. When I say horizontally, it's not mother to child, but horizontally between two living organisms. Gene could be transferred by viruses by multiple mechanisms. Commonly, we speak about the way they donate, gene transferring through the bacteriophage, but there are multiple other methods through which a virus could give a new gene or a new function as a result to a cell. It could be a unicellular organism. I will show you how multicellular organisms or complex eukaryote organisms like humans could suddenly get a new function or a new gene through a virus. So viruses seems to be contributing to the web of life and how humans have evolved and developed. So as a result of this horizontal transfer of material, genetic material, new chromosomal rearrangements happen, and you have new regulatory regions, new genes, genome-wide innovation, as in a new function suddenly appears in an organism, contributing to evolution or destruction of another species because of that. Structure could change. You might also get a deleterious effect for generations after, only for that to appear again. One of the examples that we see in humans is the occurrence of a nerve, dormant lying virus in the nerves that suddenly reactivates in the form of herpes zoster, where suddenly a gene effect has happened. Here I have shown you a cartoon where 21 chromosomes of the human body is shown, and the regions that they code for viral receptors. So viral receptors seems to be stuff that is contributed to and coded for by our chromosome itself, and that shows how we have co-evolved with the viruses in the past. For example, the dengue virus has a gene, chromosome 7 receptor, and our close friend COVID also has a viral that is coded to by one of these genes: influenza virus, Epstein-Barr virus, herpes virus at multiple sites, and so on. You could have a glance at it. When we look at the human genome, per se, we know that only 2% of the human genome has protein coding. The rest is actually that are not usually transferred to, translated or transcripted, and could actually be silent or sometimes do functions that we don't know. Actually, if you look at the whole human genome, we can also appreciate that 10% of our own genome is contributed to by viruses. Most of them because by retroviruses, because of the way they produce, because they don't do their own, they do this reverse transcriptase mechanism to introduce their own gene into our genome. And we seem to have 8% of our own genome contributed to by retroviruses and 2% by other viruses. So that's why I asked you the question at the beginning, how much of you do you think is contributed to by viruses? And here we see shocking data about our own genome, but also about the way we code for receptors that then are used by viruses for entry into our cells in my previous slide. And earlier on, I showed you the number of viruses that are harbored in our system. Actually, there is not a single place or cell that you can find absence of viruses in our human body. I want to just diverge a bit to show you this in the reptiles and in the avian species. And the one thing that these two species do is to use an egg for reproduction. So basically a yolk sac and the embryo there, which then develops independently. Now, a human or the mammalian reproduction, of course, depends on this yolk sac being dormant and the rest of the cells becoming a trophoblast to invade basically into the womb of the mother in order to derive its energy. Whereas in the egg, it is stored and used from the yolk, whereas in the humans and in the mammals, you can see that the yolk is get rudimentary structure, and the trophoblast, which derives its energy and other connections from the mother's endometrium, is the tissue that has made the difference. So in order for humans to evolve, we needed that trophoblastic structure. So how would you suddenly jump from being an egg into a trophoblastic structure producing animal or a being? And that would be by cell invasion. And that, if you successfully do, you would be able to implant the embryo into the mother's womb and as a result develop in a more secure environment than inside the eggshell. And what better organism than the virus, who are the invasion specialists, to give us that function. So if you look at the human placenta, which is a revolutionary organ that allows mammals to reproduce in a completely unique manner, you can see that the molecule called syncytin, which is a cell fusion specialist. In other words, it is the appearance of that molecule, the syncytin, in the trophoblast that allows the trophoblast to invade the endometrium and therefore implant the embryo into the mother's womb. And that is, as I suggested, would come from a herpes virus type of a gene, and that is how we jumped from being an eggshell protected organism to an organism that would develop inside a completely secure environment. Another viral gift to us for our own well-being is the P53, known as the anti-cancer molecule within us. If you have a deficiency of P53, you would be at a high risk of developing malignancy. So first of all, let's look at what P53 does. So in a normal cell, if there is DNA damage, which can cause replication of that cell or abnormal replication of that cell, you have the appearance of P53 that suddenly appears, and that risks the cellular division at that stage, and P53 will disappear if the chromosomal damage goes away or repaired, or if the chromosomal damage doesn't repair, the P53 is completely activated, resulting in that cell death because it cannot divide anymore. So P53, once again, is a virally donated mechanism within us. And I can keep talking about how we have been interacting with viruses. For example, in this graph, you can see that the crab zinc finger mechanism that is once again appears to be a defense system against viral entry into the cell and abnormal division of the viruses in our system. So we seem to have been either beneficiaries or sometimes fighting against these viruses in our past. So having spoken more or less philosophically in the past few slides, I'm just going to go to my next topic, which is to talk about hormones that are made and coded for by viruses, or in other words, viral mimicry of human hormones. So what I'm showing here is a corn snail, which is found in the Australian waters, which actually fishes. And you can see, as I show you the image, you could also appreciate the fact that it has something like an insulin delivery device, and perhaps an ideal insulin delivery device. As I call it, you can't see the needle. It is implanted within the proboscis, and as the snail attacks the fish, it So I'm not showing virus here, but what I'm showing you here is that is a snail that hunts fish by using a toxin injection system. So when a fish comes in close proximity, it injects a venom that actually has insulin. So you can, and if you study this insulin, you can see how innovative this insulin is. So this is the human insulin structure. Here you can see it has a short alpha chain or an A chain and a long B chain. The long end of the B chain is where you form hexamers with human standard insulin, but you can see that the corn snail insulin lacks that long tail. As you can see here, most of our analog insulins are modifications of this last segment of the human long chain of the B chain of the insulin. And you can see that a smarter way to do that insulin production is not to have that long tail at all. And so you have monomeric insulin produced. And from the previous slide, you saw how insulin is used as a wonderful weapon by the snail. So using a hormone as a weapon is not strange in animal kingdom. So this paper, which made me think about viruses 2 years ago, was based on this work, based in Boston and many other universities across the world. They looked at 7,455 viral genomes that were then known and asked this question: do they code for 62 known human hormones and peptides? So EMBRA, Altindis, and the others got started on this paper. And this paper was published in 2018 in the PNAS journal, as you can see. So what did they find? They found that when they matched the molecular structure, they could find similarities in viruses for many of the human molecules, including insulin, IGF, and many others, transforming growth factor beta, adiponectin, interleukin, etc. So what they do next, they didn't stop here. They went to look at the 3D structure of these hormones that would be produced by the viruses called the VIPs, virally induced peptides, and studied against the human hormones and came up with two shortlists. So they found that iridovirus, which is a fish infecting protein shown here, and lymphocystis disease virus, which causes lymphocystis disease in the fish, once again, produce these four hormones that you can see are almost similar to the human insulin and human IGF. And so you can see LCDV is the lymphocytic disease virus, and iridovirus here, Singaporean iridovirus, and they both have a 3D structure almost like a human insulin, and they also have almost similar structure to the IGF molecule in this. So they went on to show that in not just in structure, but these four molecules also were similar in their function to both insulin and IGF in the sense of causing glucose reduction and in the sense of causing cell proliferation and binding. They were almost similar to these human hormones. So what are the potential implications of viruses expressing hormone molecules that are almost similar? So this is a cartoon that explores that concept. So virus-induced protein-carrying virus. In this example, these were just two viruses that were found commonly in fishes. Infected cells start to express the VILPs and can cause endocrine signaling by producing insulin, for example, into the blood, or can do paracrine signaling, resulting in hypoglycemia of unknown significance or unknown cause without our knowledge, insulin resistance without our knowledge by producing insulin-like molecule or IGF to induce insulin resistance, and also more significantly, whether they contribute to tumor formation or acceleration of tumors by paracrine action at that site is something that the authors have suggested as potential mechanisms of these two. When they did that, they also highlighted that only 7,500 viral genome was studied, and that is all we have explored so far. And there are, as I showed you at the beginning, there are millions of types of viruses across the world. And when we come across them, and if they produce viruses, and if they produce VILPs, you can consider the potential impact of that. So having spoken about something scary, I'm going to now move on to ask another very relevant question that had been thought about even during the pandemic, is to ask why the flu hits men harder. Because we know that even with COVID, men get the infections that are more severe, almost 3:1 more severe prevalence is seen. And here you can see previous influenza, pandemic, and seasonal. You can see that men tend to have more severe condition compared to females. Is there any explanation for that? Well, here is a cartoon that is from a recent paper in the Science Journal. You can see the pink is the females and the blue is the males. So that is the estrogen effect, and this is the androgen effect. So here you see the estrogen receptor signaling results in favorable molecules like interleukin 8, interleukin 12, interferon, which is now being explored as a therapy for COVID. And their expression is what estrogen seems to induce, whereas androgen tends to induce more inflammatory molecules like tumor necrosis factor, interleukin 6, which are also associated with more severe influenza, and we also know that their expression is associated with the hyperinflammation or the severe COVID syndrome that we see. And so therefore that seems to explain at least partially why men would get a more severe infection than women. There is at least one trial that is exploring the possibility of giving estrogen to men to see whether men with COVID, that is, to see whether they would do better than usual. The possibility of a pandemic had been something that had been taught to us as med students. I remember our virology professor teaching us about a possible pandemic being imminent at any time of the year because of the way we travel, the close proximity, etc. So this paper, which didn't come out now, but during the last decade, said that the possible pandemic pathogen would be having these 4 characteristics: that it would be a respiratory transmitted virus, and they also said that it would be a virus and is likely to be an RNA virus, capacity to spread during incubation period while people remain asymptomatic, and that would be one way in which it would spread rapidly. It would be a virus that the hosts do not have immunity for or no previous exposure to, and that would be a novel virus, and that it would also be something that is very virulent in order to cause severe infection but be able to spread during incubation period. So we know that in the past we have had 2 previous coronavirus outbreaks in the 2002 to 4 during the SARS outbreak and 2012 during MERS-CoV outbreak. Both of them use the ACE receptor as their entry point. And this time we have an upgraded attachment or affinity containing virus that has caused the disease. And we know that the novel coronavirus, or the SARS-CoV-2 as we call it, has binds to ACE receptors which are ubiquitous. They are the ACE receptor seems to be everywhere in the body you look at. It's in the nasal and lung, but it's not limited to that. One of the reasons why patients get severe infection is because it's expressed in the heart, in the kidney, and now we also can see that it is expressed in the endocrine organs like the pancreas. And I will also show you in my next few slides that it's expressed in almost any endocrine organ that you look at. So a ubiquitous structure like the ACE receptor is the one that the virus enters through. And just a cartoon to show how the TIMPRESS first changes the S protein and cleaves it so that it can then bind to the, after cleaving to the ACE receptor that causes the cell to receive the virus inside. And then you have the viral RNA released from the SARS-CoV-2, which causes viral protein synthesis. And from the endoplasmic reticulum, you have the Golgi complex, and from there you have extrusion of more copies of the virus. So the process of ACE binding has been a matter of complex study. And here you have this cartoon that was published in Circulation Research in 2020 that summarizes what we know about SARS-CoV-2 and how it binds to the ACE and causes the disease. So in general, if you have ACE inactivates the angiotensin 2 and converts the pathway towards angiotensin 1 to 7, which are all protective, anti-fibrotic, antioxidant, anti-hypertrophic, and vasodilatory. What happens when the virus hijacks this mechanism of ACE2 is that you have excess of angiotensin 2 because you don't have conversion of Ang2 into angiotensin 1 to 7. Angiotensin 2 binds unaffected or in an excessive manner to the angiotensin receptor, and that causes the detrimental effects of fibrosis, increased reactive oxygen species, hypertrophic. It is also vasoconstrictive, and this process also releases more of the harmful hormones, as you can see, interleukin 6 and tumor necrosis factor alpha. And this is how the virus causes its inflammatory process. It's also, if you didn't remember, ACE2 receptor was actually discovered on year 2000 at the turn of the millennia, and we are now celebrating the discovery, 20 years since discovery, and also discovering that SARS-CoV-2, which is causing major havoc across societies, is using that system. So as I said, ACE2 receptors are ubiquitous, but what we asked ourselves was, is it found in endocrine organs? Yes, if you look, you can see that it's found in every endocrine organ, not just in the heart and the kidneys, but in pancreas, in the gonads, in the thyroid, and in the pituitary hypothalamus system. You can see that the ACE2 is expressed. Now, if that is the case, can it cause endocrine diseases is the question we asked ourselves. And yes, in the previous infection and we did a literature search to find that in the previous infection with SARS-CoV-1, as well as with the MERS virus infection, there are case reports and sometimes more extensive studies, biopsy and antibody studies to show that all these can be affected. So SARS-CoV-2, whether it could do that is our question, and our hypothesis is because it's similar, we need to look out for these problems. So we wrote this article, The Impact of SARS-CoV-2 Virus Infection on the Endocrine System, exploring its past experience and exploring the limited literature we have had ever since this pandemic started. And you can see that it's been a very popular article with more than 50 newspaper articles quoting that, and the article generator for you in my latter part of this presentation, the potential mechanisms or the potential damages that the SARS-CoV-2 could do. So first I'll start with at the top by showing you how it could affect the hypothalamo-pituitary system. Yes, in the hypothalamus, it can have direct hypothalamic effect and can also cause hypophysitis by binding to the ACE2 receptors in the hypothalamo-pituitary function in the system. We also know that anosmia is a feature of the infection, and that is because the first nerve, which is just close to this, the first cranial nerve is inflamed or infected, and the virus can enter in and can cause direct hypophysitis or can cause ACE2 binding and the reduced function due to its inflammation. So these are potential mechanisms by which patients could develop problems. Post-viral fatigue with hypocortisolism is the common manifestation of this problem in the previous infections. We know now that the post-COVID patients, there are quite a lot of them who have post-viral fatigue. At least some of them would potentially be due to hypocortisolism. So it's important to keep our eyes open and to look for this problem. We also know that the SARS-CoV-2 mimics the ACTH structure in at least 34 sites of the hormone. And so that's important to remember that it can cause ACTH mimicry, and that could be one of the reasons why ACTH producing tissues could be targeted by the body's own inflammation. Whether it actually happens or not has been a matter of debate and a hypothesis only. So if we discover more about it during this pandemic, it would be interesting. Moving on to the next important organ, the adrenal gland. Similar to the hypophysis, in the past, we have had case reports of adrenalitis due to the infection by SARS-CoV-1. We also need to remember that patients with pre-existing, we need to look for that, and they need to have aggressive treatment with steroids, etc., to be treated. We also can think of the critical illness-related corticosteroid insufficiency or koala stress syndrome, because if you look at the cortisol response of animals and compared to humans, you can see that the sheep and koalas, because they have very tiny adrenal gland, cannot mount stress response, and that is called the koala stress syndrome, because certain people with suppressed reserve of the adrenal cannot mount the response they require, and we need to look for critical illness related corticosteroid insufficiency in patients who have severe illness, particularly when they are in those who are critically ill with hypotension refractory to fluid or pressure mechanisms, or if they have hyperkalemia, hyponatremia, and hypoglycemia, usually all three together. But in all patients who are hypotensive and refractory to fluids, you might want to think about SERSI. Random cortisol less than 275 is suggested as a method to diagnose. If you give synacthen and the rise is less than 250 after an hour, that is also another feature on how you can diagnose. There are various protocols on how to treat, but the usual protocol that had been recommended for SERSI, this is without randomized controlled studies, of course, in guidelines by various societies, is to give up to 400 milligram, that's about 100 milligram in 4 times a day of IV hydrocortisone for 3 days. When the patients respond, you could give the other doses. You might also remember that only very few therapies have shown success for COVID. Interferon is one of them. And on the other hand, steroid therapy, which is dexamethasone, that had been given in a randomized manner to half of the populations in a study done at Oxford, had shown response. And whether some of them have SERSI, some of them had pituitary problem, and ACTH mimicry is very interesting to find, because as we talk about it, it's important to remember that so far only one study has been published that has shown that steroids are useful for generalized survival of patients. Corticosteroids are a two-edged sword. They, of course, can treat patients very well if they particularly have potential SERSI or have ACTH deficiency, ACTH cortisol deficiency. But also we have to remember that after a cytokine storm, there is an immune exhaustion, and giving steroids may cause further immunosuppression and can put the patient at risk of further harmful infection. So we need to remember that. So I want to move on to the other organ, thyroid. all of these can be expected. Only one has been published so far. We have several case studies from Italy that have shown that patients have presented with subacute thyroiditis. And some of the studies have, one of the latest one that was published about 2 or 3 weeks ago, showed that patients had high T4 and low TSH, as in an acute subacute thyroiditis. And they tended to be happening in patients who are really unwell. So compared to the mild COVID infection, the ones with severe COVID infection seems to have infiltration by the virus itself, causing thyroxine release into the system, and subacute thyroiditis has been reported in many case reports with this pandemic. On the other hand, sick thyroid syndrome, which we see in many patients, could be seen low TSH, low T4, low T3 due to the conversion from T4 due to the deiodinase enzyme that causes reverse T4 to be manufactured. So sick thyroid syndrome in our traditional test will present as low T4, TSH, and T3. We also could have primary hypothyroidism, which had been described in previous epidemics with the SARS-CoV-1, where destruction of follicular and parafollicular cells could occur, resulting in primary hypothyroidism. As I said, due to hypophysitis or hypothalamic involvement, you might have low T4, low TSH, which is a manifestation of central hypothyroidism. Sick thyroid syndrome mimics that almost exactly. So we need to be very aware of this potential problem. We know that with COVID, lots of patients are presenting with studies and reports from Italy in particular at the onset of this epidemic, as well as from US, that suggests that patients do present with ketoacidosis as well, and present like an acute type 1 diabetes. So these are the three possible mechanisms by which COVID could give rise to problem. And this has been seen with the previous epidemic as well. Viral injury through the ACE2 expressing beta cells, causing viral injury of the beta cells, causing pancreatitis or inflammation of the beta cells. Hyperglycemia. So in diabetes patients, there is hyperglycemia that glycosylates ACE2 molecule and the viral S protein, and that makes viral S protein enter the ACE2 more readily, and therefore diabetes patients could have more viruses into their pancreatic beta cells, resulting in more severe infection. We could also have direct cellular attack of the COVID, resulting in the characteristic pancreatitis syndrome that is due to systemic inflammation or immune mediated as well. So there are three mechanisms by which you could have pancreatitis. So in all of these, you will have insulinopenia and hyperglycemia. We also know from many studies that are coming from China and other parts of the world, we see many abstracts being submitted as well, where initial high glucose level is directly related to the outcome of the patients, and that whether that is due to expression of all these problems in the cells, and whether the degree of glucose shows you the severity of the infection, as in pancreatic involvement, is one way in which we can think about, but could also think about hyperglycemia causing more severe infection, and hyperglycemia is also generally a marker of more severe disease in other conditions as well. So there are multiple things that we need to think about to eke out what exactly the mechanisms are. But as you can see, there are multiple mechanisms by which these patients can have. At least the preliminary studies that are being sent for journal reviews from China have, and particularly from Wuhan, I have seen 2 or 3 that have come for journal publication reviews, and where they show that diabetes patients tended to have higher mortality compared to the other patients. And these are still in publication and review, so I don't want to talk more about that. In my penultimate slide, I also want to show you the importance of vitamin D. And vitamin D, as you can see, is something that is closely related to ACE2 expression and angiotensin 1 to 7 manufacturing. And in vitamin D deficiency, you could have deficiency of angiotensin 1 to 7, potentially. Vitamin D also is anti-inflammatory in the sense of reducing tumor necrosis factor. It also gives you more of beneficial in cytokines, and there are many mechanisms by which, and it also, because while it does the angiotensin 1 to 7, it inhibits the angiotensin 2 production, which is harmful. So vitamin D seems to be a key inflammation-related molecule. Whether this is playing a role in the current pandemic, in the severity of vitamin D, whether it's related to the outcomes of patients, it's still early days to think about. But lots of people are now beginning to be aware that vitamin D is a major immune modulator, and the deficiency could be harmful is something which is also thought through, because at least in UK, you see that the Black and the Asian communities, which tend to have more vitamin D deficiency, tending to have poor outcomes, whether it's just due to the vitamin D deficiency or other socioeconomic factors. They also were poorer in their socioeconomic category. So whether health-seeking behavior or the way they got it had anything to do with it, we don't know. But one of the potential mechanisms is that they were low in their vitamin D levels. So ladies and gentlemen, thank you for being patient with me. I learned as much as you would have learned. And this is my final and the summary slide. So my first point that I spoke to you about, if you remember, I asked the question, are viruses always our foe? Are they our friends as well? I showed you how they are ubiquitous. They are ecosystem stabilizers. They influence global scale ocean productivity and are responsible for the food chain to be maintained in the oceans. And they are responsible for us breathing oxygen today. And they seem to be also the virome in our body is a key player of influencer of our immunity, sometimes harmful. We don't know whether they play any major important role other than that. I spoke about horizontal gene transfer, how they play a key role in protein adaptation by driving rapid advances, sudden onset of new function in an organism, and how I showed you even in humans, we seem to be using viral mechanisms even for our reproduction and the way we mammals reproduce. I spoke about estrogen and its protective role, and estrogen as an important defender. So if you have a gender-biased society where you ask the question of females versus males, you can now decide which gender is better. I showed you the outcomes in COVID and in the influenzas on how estrogen plays an important role. I also showed data on VILPs, virally induced proteins, and how they could be potentially pathogenic. And I also showed you that we just are scratching the surface with this matter, because only 7,500 viruses have been studied. And even within that, we had very close contact with two human-like proteins in those viruses that was found among the 7,500 viruses studied. And then I finally showed you how SARS-CoV-2, the current pandemic can cause endocrine devastation. It can affect almost every endocrine gland. I showed you potential mechanisms for pituitary, thyroid, pancreas, and in adrenals, how the previous SARS-CoV epidemic had resulted, and some data is emerging as to the same occurring with the SARS-CoV-2 infection. So it's early days of the pandemic. We will keep learning. More data is to be collected. More follow-up of these patients on a long term is required as well. So I'm going to stop there. I once again thank the Endocrine Society, Sanjay, and the two chairpersons for having allowed me to speak and for inviting me. And I hope the audience learned as much as I did through this exercise. Thank you.